Good morning again, guys. Time for the video on lipids. So in this video, I'm going to cover beta-oxidation and fatty acid synthesis. I'm probably not going to cover fatty acid storage. It's a little more intuitive. So I think you guys can review that one on your own. And for the sake of keeping this video at a digestible length, I'm just going to cover those two topics. So we can start off with oxidative metabolism of lipids. And we'll go to the introduction first. So we have been talking that glucose is the primary fuel source of energy for our bodies, but fat is also a major fuel source of energy. And we're going to see why when we talk about how many ATP we can get from one fatty acid. So we're going to tend to use our glucose first, but usually our glucose, when we need a lot of energy, is not going to be enough. And that's when we can start to break down our fats to produce a lot of energy. It just takes a little more effort and a little more time, and that's why we don't just start with fats. So fats we usually see as triglycerides. So remember, that's like our glycerol backbone. I'm just rough sketching. This is not accurate, but and then you have your fatty acids attached to it. So that's your triglyceride. Does that mean that all fats are packaged like that? No. It just is the most common packaging. And then when we break something down, that's called catabolism. We also have anabolism, which is the building up of molecules, but this time we're focusing on breaking them down. And the entire process of breaking down fatty acids is going to be oxidative. Don't panic, you know what this word means. Remember our redox reactions, where we reduce something, and reduction involves gain of electrons, and oxidation involves loss of electrons. That probably makes a little bit of sense if we think about it. We're going to be breaking them apart. So when it loses some of its atoms, it's going to lose some of its electrons. And we're going to see as we get into the meat and potatoes of beta-oxidation that we're going to be breaking bonds at something called the beta carbon. So we call this process beta-oxidation. So these are just kind of a review of fatty acids. Remember, they come in different forms. Unsaturated, no double bonds, only single bonds. We can have saturated fatty acids where there are double bonds present, and they also come in two flavors, where we have trans, making this nice straight pattern, and cis, where we get more of a curved feel to it. Our trans fatty acids are our unhealthy ones. And what trans and cis means is what side the tails on either side of the double bond are on. So you can see we have one going up here and one going down here. So trans means different sides, versus cis, which means the same. See how both bonds are pointing down? We got our tails on the same side of that double bond. This is an overview from Biochem 1 of what the different fatty acids are, or the different fats are, how many carbons they have in them, if they have any double bonds. Remember, this one should have no double bonds because they're saturated. This one does have double bonds because they're unsaturated, and that tells you how many double bonds there are. This tells you information about where those double bonds are. So here's a great example. 18 carbons, 3 double bonds, and this is the location or the number of the carbons where those double bonds are located. This is our delta naming system. And this is the common name we give it. That's alpha-linolenic acid. That's one of our essential fatty acids. The omega name of that is omega-9, because that's where that first double bond is. I did ask him. He said There is no value in rememorizing all of this. This is just to refamiliarize ourselves with how we can name and talk about fatty acids. How do we digest them? We're going to eat them. I probably eat way more fats than I need to. They're going to come in different forms, but primarily triacylglycerol, in the form that we were talking about earlier. We're going to chew it up. We're going to swallow it. In our mouth, we're going to start with an enzyme called lingual lipase. We talked about salivary amylase with carbs. Lingual lipase is a similar working enzyme, but it's just specific to fats. Then we get into our stomach, and now we're going to have gastric lipase, and that original lingual lipase will be denatured. So now gastric lipase is doing the work. And then from the stomach, we're going to reach the small intestines. This is where the most important action happens in almost all food digestion. We're going to use bile from the gallbladder and the liver to do the true breaking down of the fatty acids. These two sets are going to more chunk them up into reasonable sizes if they're really large. This is when we're really going to start breaking them down. More of that. Remember that when we take in dietary fats, we're going to package them in a little bus called a chylomicron. And that chylomicron will take those fats wherever they need to go throughout our body. We have other types of lipid packaging systems. Remember LDLs, HDLs. Those are more for our endogenous fats, depending on their size. But when it comes to dietary fats, we talk about chylomicrons. So this is a quick overview. I'm not going to spend too much time here, but when we take in fatty acids to do the beta oxidation, we have to get them into the mitochondria. So the mitochondria is where the action is happening, but we know by now that we can't just waltz into the mitochondria. We've got to have, like a ticket first, or some sort of process that gets us in there. So this is telling us that we're going to have to do something called activation of our fatty acids. And then after they're activated, we can use a carnitine shuttle to get it into the mitochondria. And we're going to go into more detail in a few minutes. So this is just telling us that beta oxidation, like I was just saying, is primarily going to occur in the mitochondria, but there is an exception. We sometimes have very long chain fatty acids, and that's exactly what it sounds like. Those chains that we see are just very, very long, greater than equal to 22 carbons. They are not going to go to the mitochondria. They're going to go to peroxisomes. So peroxisomes, think back to cell physiology. They're an organelle that essentially has hydrogen peroxide in them to help break down substances in our cells. So this is going to be where our very long chain fatty acids go. Think of this as an exception to the rule that fats are broken down in the mitochondria. This is saying the same thing. The only new information is how these fats can get across the membrane. And what you need to know is short and medium is some sort of diffusion. So free diffusion means it's going directly across the membrane. Diffusion means there's probably a channel or a carrier involved, but there's no energy or changing of the molecule involved. And then the long chain, we're going to talk about the carnitine cycle. This is where that carnitine shuttle I had mentioned previously is going to come into play, getting us into the mitochondria. We know that our very long chain fatty acids need to go to the peroxisome. We don't really know how they get there. So beta oxidation primarily is going to occur in the liver and the muscle. It's almost always going to be the liver and the muscle. They're going to produce the same things: acetyl-CoA, FADH2, and NADH, and an H+ ion. So different location, same product, but what we do with those products is going to change based on our location. We're always going to produce acetyl-CoA, but what are we going to do with it? In the liver, we're going to make something called ketone bodies, which are going to be sent out of the cell, And that process is called ketogenesis. Genesis means the making of. Keto refers to ketone bodies, so liver, ketogenesis, making ketone bodies. Acetyl-CoA, we know, can go into the Krebs cycle. So any acetyl-CoA made in the muscle is going to go into the Krebs cycle to make ATP. So to get fat out of our adipose tissue, I'm starving, I haven't had a meal in a long time, we're going to use a lipase, an enzyme that breaks down lipids, to chop up our adipose tissue. This is hormone sensitive, and we're going to see why. And note that it needs normal thyroid level. We've talked a lot about the thyroid in the systems physiology. Another reason why it's important is for breaking down fats. This is why when you have a thyroid disease, you tend to have lots of keeping on or keeping off of weight issues. And when we do break those fats off of the adipose tissue, they can't just walk around the body on their own free will. They're going to need a special chaperone called albumin, and that can carry 6 to 8 fatty acids at a time. So that's just going to guide it to where it needs to go. So now we're going to get our fatty acids into our cell. We've been talking a lot about what fatty acids are, how do we start breaking them down. Now we have our fatty acid. We know we need to get it into the mitochondria. How are we going to do that? I said we have to activate the fatty acid first. To activate the fatty acid, we're going to need an enzyme called a thiokinase. Remember, kinase is going to be moving phosphate group. So we're going to look for that phosphate group. This diagram shows it as 2 separate steps. It's pretty accurate to say it that way, but if you're getting confused, what you're seeing in this picture is the same thing, they just have it all condensed. So this is just breaking it up into digestible pieces. We're going to take our fatty acid and we're going to take some ATP. Remember, breaking down fatty acids is going to produce lots of ATP, but we've seen in a lot of catabolism processes, we sometimes need to invest a little ATP in to start. Same thing that's happening here. So when in glycolysis, remember we had to add 2 ATP in the beginning to get a net 2 ATP in the end. We're going to add a little ATP in the beginning to get lots of ATP in the end. So that thiokinase is going to help us take a phosphate group off of the ATP and add it to the fatty acid. So here's that fatty acid, same thing as right here, and here's that phosphate group attached to it, and we're going to leave the adenosine on it too. So the adenosine is the A of ATP. So this is like its intermediate phase. The same enzyme as before is now going to take a coenzyme A. So if you see CoAsh, that's coenzyme A. And it's going to do a little replacement action. That's still the same fatty acid before, and S-CoA is coming from that coenzyme A. And you're going to see that it's going to release AMP. If I look here, I had adenosine and a phosphate group. We know adenosine with a singular phosphate group is AMP. That's what we're seeing right here. So this whole process, we did move the phosphates, that's why it's called a kinase. But we took a fatty acid, and the end product is something called an acyl-CoA, which is a fatty acid with a coenzyme A attached to it. So that's exactly what's written out here. Okay, so now we've activated the fatty acid. Now we're actually going to get it across the mitochondrial membrane. How are we going to do that? We're going to use the carnitine shuttle. I'm going to use this picture to explain most of it, but all of this is just writing out exactly what's happening. Take a minute to read through it. There's actually more than one enzyme involved in this. They're going to be called CPT1 and CPT2. Which stands for carnitine palmitoyltransferase 1 and 2. They're both part of this carnitine shuttle. They're going to do just slightly different parts of it. Take some time to understand what carnitine is. I'm sure he's going to ask a question from right here. But what's happening is we have this activated fatty acid that we have in the cytosol, or the cytoplasm of the cell, and we need to get it into the mitochondrial matrix. We know in between the cytosol and the matrix we have this intermembrane space. So we actually have two membranes to go through, so that's how you're going to remember that we're going to need two enzymes to do it. So the first enzyme is, they have different names here. They have carnitine acetyltransferase. We're going to say it's the same thing as carnitine palmitoyltransferase. The difference here is this one is going to be specific to palmitoyl. This one is just a more generalized version. You could see it either way. And it's going to take this activated fatty acid and a carnitine. It's going to mush them together. It's going to spit out that coenzyme A. This coenzyme A is going to be reused. And now, instead of that coenzyme A attached to our fatty acid, we have a carnitine attached to our fatty acid. That's going to allow this carnitine fatty acid into the intermembrane space. So that's membrane 1 crossed. Now, how are we going to get it out? Well, the carnitine fatty acid can come out through a special transporter, but we don't want carnitine fatty acid. We want a acetyl fatty acid, or an activated fatty acid. So this is when we need our CPT2. It's going to take our carnitine fatty acid, and it's going to take the carnitine out of it and replace it with that coenzyme A again. And here we have our activated fatty acid, just like before. And you heard me say over and over again in my last video that whenever we take something, change it, move it, and change it back, it's because that original molecule can't go through a membrane by itself. And that's what we're seeing again with the carnitine shuttle. We saw it with the malate shuttle, just another shuttle system specific to fatty acids. So now we have the fatty acids in our mitochondria. How are we going to break them down? All right. So here's a long picture that's going to outline the process. I'm going to skip it until we have a little more background information. So beta-oxidation, 4 steps. I think they're pretty simple. 4 steps means 4 enzymes. I'm not going to spend a lot of time on this slide because I think I said I was going to skip this. I'm not even coming back to it. I think there's a better picture to diagram it. Before we diagram it, we just need to talk about the products a little bit. So you're going to have a fatty acid. You're always going to split it up into 3 products: acetyl-CoA, FADH2, and NADH. We saw that before. We can use the number of fatty acids to determine how many, the number of carbons in the fatty acid to determine how many of each product we're going to get. It's always going to follow a pattern. Here's the equations for those patterns. So I'll just run through one example. Palmitic acid has 16 carbons. 16 divided by 2 tells me I have 8 acetyl-CoA. I can do 16 again. 16 divided by 2 is 8 minus 1. I'm going to get 7 FADH2. Same as NADH. I did ask him about this too. It doesn't look like we're going to have to do any of these calculations on the exam, or he would at least give us a heads up if we do. But it's kind of helpful to know that we're going to get half the amount of acetyl-CoA as we are carbons in our fatty acid, and that's going to help us to talk about how much ATP we can produce from a specific fatty acid. Beta-oxidation. So we're going to use palmitoyl-CoA as our starting place. So what you see here is that acetyl group, that ketone group. So this is our functional group. And so just as a side note, when we name the carbons in a fatty acid, the carbon after the after the ketone group is going to be the alpha carbon, and the carbon after that is going to be the beta carbon. This is, I mentioned earlier, why we get the name beta-oxidation. This is where it's going to become important. So we're going to watch where bonds get broken, and you're going to see it's going to be between the alpha and beta carbon. And then this is all just demonstrating the long carbon fatty acid chain after the alpha and beta carbon. So enzyme number 1 is acyl-CoA dehydrogenase. You're going to see if I go back here, acyl-CoA dehydrogenase. So this slide you can match up to this picture very easily. We're going to use FAD. We're going to oxidize this palmitoyl-CoA. That's going to give me FADH2. And when I oxidize palmitoyl-CoA, all the action is going to happen in here. So I'm going to start by forming a double bond between these carbons. That's what you're seeing right here. So remember, when we oxidize something, something else is being reduced. Reduction involves gain. That's what you're seeing right here. And anytime we form one of our electron carriers, we can take it to the electron transport chain. Remember, we're trying to produce energy. A lot of energy can be produced at the electron transport chain from the oxidizing of molecules like FADH2. So that's what this is representing right here. All right, so now we have our double bond. So now we're going to use an enoyl-CoA hydratase. Hydratase, think hydrate. We're going to add some water. Water is good for making OH groups, and there's an error in this. This should be this. But we added that OH group. And so now we don't have a double bond, we have a single bond, but instead of hydrogens, we have an OH. So next step is going to be beta-hydroxyacyl-CoA dehydrogenase. Dehydrogenase, you know you're going to have a redox reaction. We're always oxidizing the fatty acid, so we're going to have to reduce something else. Instead of FADH, like we saw up here, this time we have NAD. So that NAD is going to produce NADH when it gets reduced, and that can go to the electron transport chain to make more energy. When I oxidize this molecule, I'm going to oxidize it right here, and that's going to give me a double bond. You should see 2 ketone groups right now. The whole goal of step 1, 2, and 3 is to form another ketone group at that beta carbon. Next, we're going to have an acyltransferase, or you can call that a thiolase, and it's going to use another CoA or a coenzyme A to break the bond between the alpha and beta carbon. So what we're seeing here, this came from this, and this came from that, and we stuck that on the end of it. So this right here is acetyl-CoA. That is our first of many acetyl-CoA we can pull off of this fatty acid. So acetyl-CoAs have 2 carbons. I just broke off 2 carbons from my starting molecule. That acetyl-CoA is going to go through the TCA cycle. You're going to get lots of ATP from it. That's great. Not only are you going to get ATP from it, but this 10 is coming from also the electron carriers that are being produced from the TCA cycle alone. So essentially what we have here is something very similar to what we started with. It's just 2 carbons shorter. 10 here, 12 here. We're going to take this and we can do it all again and produce a bunch more ATP and another acetyl-CoA to go into the TCA cycle. So we're going to chop off carbons in groups of 2 to form acetyl-CoAs to break down this fatty acid. And so it's really just a 3-step process of forming a new ketone so I can separate it again and we can have another ketone to start with. Does that make sense? We have a ketone here. I added a second ketone so we can break the bond here. That's all. I'm going to skip ahead. This is bringing us back to the tricarboxylic acid cycle. We're talking about this part right here. So when we break off these acetyl-CoAs, they can go through the citric acid cycle, and that's going to produce a lot of ATP on its own. And so we have just a bunch of slides here telling us we can go through the or the tricarboxylic acid cycle, and the electron transport chain. And essentially, between this and that and all the electron carriers we produced when we did our beta oxidation here and here, we're going to produce 106 ATP in one palmitate molecule. So let's go back to here. Palmitate, it's almost the same thing as palmitic acid. So from a 16 carbon fatty acid, I produced 106 ATP. This is comparing it to a glucose molecule. That has 6 carbons. If we break down glucose, we go through the TCA and the ETC for one glucose molecule. I'm only getting about 38 ATP depending on the source that you cite. This is a huge difference. But remember, glucose is the preferred fuel source for the body. Why? Why wouldn't I want to produce this much ATP? Well, first of all, you might not always need that much ATP. And second of all, the making of this ATP involves a lot more work for the cell. And then the cleaning up of the process that we're about to get into is also a lot of work. So we get fewer ATP breaking down a glucose molecule, but it's a little bit easier, and that's why we're going to use that as our primary fuel source. When we run out of glucose, we've talked about glycogenolysis, and we've talked about gluconeogenesis. Those processes need energy. We can get the energy from beta oxidation as well as some energy just to be used. And so let's talk about the whole cleanup part. We said a long time ago in this video that when we do beta oxidation in the liver, instead of going through the TCA cycle, we're going to take those acetyl-CoA and we're going to go under this process called ketogenesis, or the making of ketone bodies. So here are the ketone bodies. So we have 3 different options of things to make. All of them can be used to provide energy, especially in areas like the brain. So ketone bodies, we can break them down throughout the body. I think of them most in the brain because the brain just needs so much energy. It will use anything it can get, and ketone bodies are one of the few things that can get into the brain. So we have acetoacetate, beta-hydroxybutyrate, and acetone. Acetone is not that good. We're going to produce some of it. Our body can handle the normal amount that it produces, but too much acetone is very bad. Skipping ahead a little bit. So here's the process. We're taking acetyl-CoA from beta-oxidation, and we're going to make it into acetoacetyl-CoA. So that's essentially 2 acetyl-CoAs put together. And then we're going to use our HMG-CoA synthase to produce this molecule, HMG-CoA. This is like a lot of words. It's a little overwhelming. But what we're doing here is we're getting ready to export these ketone bodies. Our liver doesn't want anything to do with them, but tissues like our brain do. And we need to change their shape. And so these enzymes right here are going to catalyze some reactions that are eventually going to get us to acetoacetate. That's one of our ketone bodies. How do we get the others? One more enzyme, beta-hydroxybutyrate dehydrogenase, that can break that down further. We like this molecule a lot because we can use it to recycle NADH+. That's what's happening right here. So if we have lots of NADHs that aren't going to the electron transport chain because we have enough energy, we need to recycle them so they can be used again. So that's the point of making this. Unfortunately, when we make this, we make a little of that too. And that's what we're seeing here. When we break down our fatty acids, we're going to see the buildup of ketone bodies. We're going to see more of them the hungrier we are. So now we have our ketone bodies, like hydroxybutyrate. How do we break them down? Well, we're going to go back one more step. Remember how we did this? Well, we need to go back up here because we need acetoacetate, really. Once we get our acetoacetate back, we can use this enzyme, succinyl-CoA, acetoacetate-CoA transferase, what a long name, and a thiolase, and essentially we're going to get back to acetyl-CoA for the TCA cycle. So the whole point of making ketone bodies is for transport. This is why I like the brain as an example. We can't necessarily take acetyl-CoA and put it in the brain, but we can take a ketone body, put it into the brain, make it back into acetyl-CoA, and use that to make energy. So skeletal and heart muscle, we like it for, and of course, like I said, the brain. And then, of course, anytime we have any process, we're going to have to regulate it. The best way to regulate most processes are via hormones, and these hormones we've seen a million times before. They're going to keep coming back. Glucagon, it's meant to raise blood sugar. If we need to raise blood sugar, we probably need more energy. So we're not necessarily dealing with adding glucose to the blood now, but we do need energy to make that glucose to add into the blood. If I need energy, I need to break down fats. If I need to break down fats, I have to undergo lipolysis. This is just the breaking down of fats part. This isn't the beta-oxidation part. This is that, getting the fats out of the adipose tissue part. So glucagon is going to help activate this. We're going to see it during fasting and starvation. We also can do this under stress. This is why I'm staying skinny without going to the gym. I'm just always stressed, so I'm activating lipolysis. I have epinephrine as a stress response and also hypoglycemia, but mostly stress response, and cortisol due to chronic stress. It's going to activate lipolysis. It's going to increase the breaking down of fats for energy. Moving on, so we broke down our fats for energy, but sometimes we have plenty of energy. We're eating fatty foods. What does our body do with them? We're going to store them, and this is going back to prehistoric times when we didn't know when our next meal was coming, so any excess food or energy source our bodies found, they wanted to store it, so if we were starving, we'd have energy to keep us going. This is why you don't just die after one day of eating. It takes a little while because your body has backup storage forms of energy. I'm not recommending starving yourself, though. So, de novo is new. So new synthesis of fatty acids is going to occur in these different organs, mostly the liver. The liver does everything, right? And one reason we might do this is if we're eating too many carbs. We can make them into, so we can eat too many fats. We can obviously store the fats. We can also do this with carbs. If we have too many carbs, we can make them into fats. Protein does not get turned into fat. So two big steps in the process, acetyl-CoA carboxylase and fatty acid synthase. We're going to see those. Of course, we're always worried about location. It's going to be in the cytosol this time. So we broke them down in the mitochondria. We're going to build them up in the cytosol. Once again, if we need to transport things through a membrane, we often need some sort of system to help us do that. We're going to see the malate shuttle come back. So acetyl-CoA from the mitochondria is going to enter the cytoplasm via a malate citrate antiporter. And that's what we're seeing, right? We see the citrate coming out here. We see the malate going in here. So if we broke down fatty acids and got acetyl-CoA, that means I can probably stick acetyl-CoAs together to make a fatty acid, right? So the first part of transporting these acetyl-CoAs out into the cytoplasm is an antiporter. What's an antiporter? It's a different form of active transport. It's going to push things against its concentration gradient. But instead of using ATP to power it, we're going to move two substances at once to do that. So by pushing citrate out, I'm going to move malate in. It's the simplest way I can describe it. So now we have citrate out of the cell. Citrate can be converted into oxaloacetate and acetyl-CoA. This reaction should feel familiar because when we studied the TCA cycle, we added these together to make citrate. So instead of citrate synthase to go the other way, we're going to have ATP citrate lyase. Now that I have OAA, I can convert it to malate using malate dehydrogenase. That's the same enzyme as the TCA cycle. We're just doing it in a different place for a different reason. And now that I have malate, I can transport that and make it to pyruvate when it enters the mitochondria using malic enzyme. So at this point, this is just explaining that anti-Porter system, because there's more than just shoving malate in here and getting citrate out. But we haven't actually talked about the acetyl-CoA being used to make fatty acids. We've just produced it right here. So this is the acetyl-CoA entering the cytoplasm. The rest of this is just the anti-Porter part. All right, now we have our acetyl-CoA. So now we're going to do our stage 1, acetyl-CoA carboxylase. That's what we were talking about right here. Acetyl-CoA carboxylase. Carboxylase uses carbons. They're going to come from CO2. So what's happening here is we're taking an acetyl-CoA plus a carbon dioxide, and we're making something called malonyl-CoA. So we did extensively do this in class. You can use my color system to help you. We have pink, yellow, and green. Now we have green, pink, and yellow. So we didn't add anything. I know, like he said, it does look kind of crazy. It changes shape a lot, but we just stuck this to this. That's all. Now we need to activate that. So this is going to use insulin. I think this is probably the big takeaway from this slide, is that insulin is going to help activate this process. Citrate's going to be important in making this right here. And it's going to be upregulated by a high carbohydrate, low fat diet. So when I'm eating lots of carbohydrates, I'm going to store the excess carbohydrates as fat. So I need to increase the process of making fatty acids, and that's why a high carbohydrate diet is going to activate acetyl-CoA carboxylase. And you're going to ask me, why not with high fat diet? Well, we studied, when we did beta-oxidation a little bit, we talked about how a high fat diet is going to change your body's primary fuel source. You're going to start to undergo what they call the ketogenic diet. So that should be with a high fat diet. So yeah, high carbohydrate, we're going to need to store them as fats. We're going to need to speed up this process and activate acetyl-CoA carboxylase. If I need to inhibit it, it's going to be the opposite. So the opposite of insulin is glucagon, right? And glucagon and epinephrine go together. Opposite diet, high fat, low carbohydrate. And here's where I was trying to go to before, ketogenic diet. High fat diet, ketogenic, we're not really going to be storing fatty acids. We're going to be breaking them down. All right, so that was all stage 1. Stage 2, the actual making the fatty acids. So before we were just collecting acetyl-CoAs and kind of activating them by forming malonyl-CoA. Now that I have my malonyl-CoA, I can use that as my true building block for a fatty acid. So we're going to need lots of enzymes. It's going to have 7 enzymes in it. We're going to see an ACP, or an acyl carrier protein, because we don't like to leave molecules unattended in the cytoplasm. This is going to hold its hand and stop enzymes from breaking it down because they aren't sure what it is. And this is just talking about its structure, which you can see in this picture. We have like this, and then this is like the opposite of it. So that's why it's called a dimer. Dimer means 2. So here's the actual fatty acid synthase. This is our enzyme, and we're going to see that acetyl-CoA attached to it right here. It's not the same one as before, but we do need to always start with acetyl-CoA. Here's where our true building block, like I said, is malonyl-CoA. So this enzyme kind of gets more or less activated with acetyl-CoA, and it's going to take that malonyl-CoA and grab onto it. Now, the next enzyme in the process, ketoacyl synthase, is going to take this enzyme, and we're going to add a methyl group right here. So we see same. This carbon's the same as that, double bonded oxygen, but instead of an oxygen here, I have a methyl group now. Where did I get that methyl group from? Here. See how this is shorter? All right, moving on. I'm going to break that double bond. We're going to get an OH. Now that I have an OH, I can form a new double bond. So we saw the double bond up here. I need the double bond in here. We always have to have OH when we're moving double bonds. Now that I have that double bond formed, maybe I don't want a double bond, so I'm going to use an enol reductase to reduce that bond. So now we see a single bond. Now after that, we can stick some of these carbons up on that acetyl-CoA, and now we have a fatty acid. I'm going to let you sit, think about this, think about what we just talked about with beta-oxidation, where we broke a double bond, formed an OH, made a ketone, right? This is the exact opposite. We started with the ketone, we made an OH, and now we have a double bond, and then we got rid of that double bond, of course. We're essentially going backwards from beta-oxidation. Here's the overall reaction if we wanted to make a palmitate molecule, so a 16-carbon fatty acid. This is the same fatty acid we used in beta-oxidation as an example, where we made 8 molecules of acetyl-CoA, so now it makes sense that we're going to use 8 molecules of acetyl-CoA to make it. So this is the same thing that we were talking about before, just in a different part. So before we were talking about the activation of the acetyl-CoA carboxylase. Now we're talking about the regulation of fatty acid synthase, but we're going to see kind of the same pattern of high carbohydrate, low fat diet is going to activate it. High levels of circulating free fatty acids, that means we have plenty of fatty acids. Why would I want to make more? So that's why this is going to inhibit it. Okay, so now we've made fatty acids. There's only so many fatty acids our body can actually make. But what about if I want to make bigger ones? I can elongate, make them longer, my fatty acids using more enzymes. This time we're going to be in this smooth endoplasmic reticulum. So we know from cell physiology that the smooth endoplasmic reticulum is important for processing fats. Here's why. If we need to make a long-chain fatty acid, I'm going to start with a fatty acid I can make in the cytoplasm like palmitate and elongate it to make a long-chain fatty acid. And that's what we're seeing here. This looks long and complicated, but we can break it down a little bit. Essentially, we have the same process as before, and we are going to essentially do the reverse reaction of beta-oxidation. So we're just using different enzymes to do it. I'm going to use this picture. We have a malonyl-CoA and that palmitoyl-CoA that we made during that fatty acid synthesis stage, and we're just mixing them together. And now I have 2 ketones again. What do I got to do? Get rid of that ketone. So through a series of reactions, we're gonna, sorry, I got distracted by this. Essentially, what's happening is we are adding 2 carbohydrates, or it's not 2 carbohydrates, 2 carbons on at a time. So we're taking our original palmitoyl-CoA, adding a malonyl-CoA, and we're essentially adding 2 new carbons to it to make it a little bit longer. So we went from palmitoyl-CoA to stearoyl-CoA. And that's the same thing that they're showing here. Same enzymes, just a different format. So that was how we make long-chain fatty acids. What about medium and short-chain? We're, instead of being in the smooth endoplasmic reticulum, we're going to be going back to the mitochondria and doing the reverse process of beta-oxidation. And then how about very long-chain fatty acids? This time we're going to the brain, and we're going to take that stearoyl-CoA that we made in the smooth endoplasmic reticulum, and we're going to just add more carbons to it. So we can make fatty acids in the cytoplasm. If I want them longer, I'm going to take them to the smooth endoplasmic reticulum and use malonyl-CoA to make them 2 carbons longer at a time. If I need medium or short chain fatty acids, I'm going to take them to the mitochondria and use acetyl-CoAs to make them medium or short or whatever length I need. And if I need very long chain, I'm going to take what we made in the smooth endoplasmic reticulum, and inside the brain is where I'm going to be producing them. I'm going to just elongate that sterile CoA even more. And this is important for myelination. So a couple different locations we can be in. So we're always starting in the cytoplasm. The length of the fatty acid is going to determine where we go from there. Okay, so we've only talked about the making of saturated fatty acids. Remember, saturated fatty acids had no double bonds, but not all fatty acids that our body produces need to be saturated. Maybe I want some unsaturated fatty acids. So we're going to start with our non-essential fatty acids. So non-essential fatty acids are fatty acids that our body can produce on its own. So what we've been talking about before are non-essential fatty acids. I synthesize them from new. I didn't have to consume them to get them in my body. If I want to desaturate them, if I want to make unsaturated fatty acids, I am limited to one enzyme, and that is a delta-9 desaturase. And that's because we can only add bonds at the delta-9 carbon. So it doesn't matter what length we start with, I can only add the double bonds in one location. Since there's only one location I can add them, there's only one enzyme I can use to desaturate my non-essential fatty acids. So here's an example. We can start with stearic acid, which our body can make. If I want to desaturate it, the only place I can do is the delta-9 carbon using that one enzyme we have, and that's going to make oleic acid. How about our essential fatty acids? So these are fatty acids our body cannot produce, so it is essential that you consume them. Our essential fatty acids we've learned last try are omega-6 and omega-3, which are linoleic and alpha-linolenic acid. But I can consume them, and I can change them from there. How am I going to do that? Well, it's going to vary, and you're going to see a long list of what we can get just starting from these essential fatty acids, but they're always going to have the same starting place. So before, with the non-essential fatty acids, we only have one enzyme. It's like one and done. Here, a couple enzymes can be used, but we're going to always start in the same place, and we're going to see a pattern. Let's look at that pattern now. So starting with the omega-6 family, that was one of our essential fatty acids, right? Linoleic. Starting place is always delta-6 desaturase. So what we see here is we consume that linoleic acid. This is the structure of it when we consume it. I'm going to add another double bond. So that's why the number goes from 2 to 3. Delta-6 tells me where that bond is. It's at the 6th carbon. All right, now I can elongate it from here. Remember, I had to consume it as a starting place, but now I can change it. So elongase, we're going to add 2 carbons at a time, always in the synthesis of fatty acids. So we're going to go from 18 to 20. We still have 3 double bonds, but you see the location of them change, and that's because of where we added those 2 new carbons. They essentially just bumped these double bonds over 2. The double bonds didn't actually move. It's just the numbering of our carbons changed when I added 2 more carbons. So that's going to give us DGLA, which is dihomogamma-linolenic acid. So after I elongate, I need to desaturate again. So carbons stay the same, but I'm going to add another double bond. Where am I going to add it? 3 spots over at delta-5. So I know I'm going to have to name it delta-5 desaturase. So there's 2 ways to think about it. The name of the desaturase enzyme is always going to go down by 1 number. We went from 6 to 5. Or if you can keep track of where these double bonds are, they're always placed 3 carbons away. So if my last double bond was at delta-8, the new double bond has to be at delta-5, therefore a delta-5 desaturase. So that was like an example from the omega-6 family. So starting with linoleic acid, we can desaturate, elongate, desaturate. Let's do it again, but with the omega-3 family. So omega-3 is alpha-linolenic acid. That's an 18 carbon fatty acid with 3 double bonds. Notice how they're all 3 carbons apart. This is different from this one because of having 2 double bonds. Always, always, always start, no matter what omega essential fatty acid you have, always start with delta 6 desaturase. What does a desaturase do? It's going to add a double bond. So, number of carbons stayed the same. We went from 3 double bonds to 4. Where is that new double bond? It's at delta 6, which was 3 away from delta 9. After I desaturate, I'm going to elongate. What happens when I elongate? I add 2 more carbons. The number of double bonds doesn't change, but those locations are all going to change by 2. They're going to get bumped up 2. After I elongate, I have to desaturate again. When I desaturate, the number of carbons stays the same, but I increase the number of double bonds. They're going to be placed 3 carbons away from the last one. 8 minus 3 is 5. That tells me the name of this desaturase is delta 5 desaturase and where that double bond is going. Always after we desaturate, we're going to elongate again. So we're going to add 2 more carbons. 20 plus 2 equals 22. We're going to keep the same number of double bonds, but they're going to get bumped over 2. Okay, I thought I was going crazy for a moment, but I did do some fact checking. There's just some typos here. This is what I found. Here's where all the double bonds are. So from that, we can tell that there's some typo. What's happening here is the rule still stays the same. Because I elongated, I added 2 more carbons. Everything has to get bumped up to 5 plus 2 is 7. 8 plus 2 is 10. 11 plus 2 is 13. 14 plus 2 is 16. And 17 plus 2 is 19. And then that typo just led to some more issues when we desaturate again. But focus on your rules. When we desaturate, we're not changing the number of carbons, we're changing the number of double bonds. And I'm going to add them 3 carbons away at delta 4, which gives me my name, delta 4 desaturase. So as I said at the beginning of the video, this is where I'm going to stop because I think this has been long enough. And I think this stuff is not too bad. I think if you guys just read and use some of your brain cells that I know you guys all have, hypertrophy versus hyperplasia, it's all easy to digest in my opinion. Have a great rest of your weekend. Good luck on the upcoming radiology quiz, and finals are right around the corner. I know it's scary, but good luck.